from MicrobeTV. This is office hours for Wednesday, October 22nd, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can learn all about viruses from me, a virologist who has worked in the field since 1975. Wow, that's 50 years. Long time, considering I'm only 50. How could I do it? I guess it's time travel. Welcome, everybody. Good to see you again. We have a fun-filled evening again of news and mini lecture and your questions, of course. Let's welcome our moderators. We have Les here tonight from California. We have Vanity Nutrition. We have... who else is here? I think that's it for now. Tom Steinberg is here. Thanks for coming week after week and moderating, keeping things civilized, right? And thank you all for coming once again to hear a little bit of science in a country that seems to want to ignore it. Tom asks me, oh, this is a good question. I'm going to just start with this. Does PVR 106 ring a bell? Of course it does. It's my full-length poliovirus plasmid. So it's a full-length poliovirus DNA, a DNA copy of the RNA genome cloned in PBR322. And that is what I put into cells in culture and found it to be infectious. That was back in, well, we published it in 1981. One of two papers I published with David Baltimore, may his soul rest in peace. We're having there is going to be a memorial for David in Pasadena in a couple of weeks, and I'm going to be going to that to pay my respects for somebody who made a big difference for my career. So tell us where you're from. We will shout you out in the first minutes here of office hours. Dagfinn is here from Oslo. 9C and raining 2 a.m. Brave to be here at that hour. I appreciate it. Not sure I could stay awake at that hour, but glad to have you. Tona is on her way home, currently at LaGuardia Airport. Let's see what the weather is here. It's getting a little bit chilly. I was in Princeton, New Jersey today, working on the Virology textbook. Here it's 11 degrees Celsius and partly cloudy. I spent Thursday, and part of Saturday in Blacksburg, Virginia, home of Virginia Tech, where I gave the keynote lecture at a symposium at the Center for Viral Zoonoses and Pandemic, whatever. I can't remember the name of it, but it's a center in the university, and it was really great to meet lots of people. Okay, Martha is in Charleston, South Carolina, where many thousands turned out for No King's Day. No kings. We don't want any stinking kings in this country, okay? And if you're not disgusted by the stupid videos that Trump and Vance released, animated, AI-generated, of them in king robes flying over the U.S. and dumping poop on the protesters, if that doesn't just turn you off completely and remind you of fraternity cranks, I don't know what would. Everybody needs to be repulsed by that kind of behavior. It's just childish, and it just represents the kind of people that are in charge of this country. Oh, my gosh. Oh, my gosh. Questella, welcome. Nice to see you. Les really likes the Beyond the Noise charts of vaccine preventable deaths, how Kennedy changes the scale of the numbers to hide vaccines save lives. Perfect example of how to mislead with stats. Yeah. Vaccines, folks, save lives. There's no hiding. There's no question about it. The numbers speak. You cannot change the numbers. And Les says they should teach this as an example in every medical school. Who else is here? So Les tells us who's going to read Steven Pinker's How We Know What We Know. Cool. I have a book that I'm reading right now, which is highly unusual. I'm going to tell you about it in a moment. Pete is here from London. Welcome. Vanity's reading Nobody's Girl by Virginia Giuffre. And yeah, the names, this is the way the names are going to be. It's your actual handle, the @ handle. It's not your actual name that you would have. So it's nice if you can change your @ handle to something recognizable. But Pete Glass is there. That's fine. We don't care about the 3496. Will is in Changsha, China, 13C, and sunny. Hope to visit China next year. I'm trying to arrange a trip with XJ Meng is at Virginia Tech, good friend of mine, and going to try and arrange a visit to a couple of virology centers in China. Kang is in Chicago, where it's 9C and cloudy, definitely getting colder, that's for sure. It's now the end of October, another week left. And before you know it, this year's going to be over. And before you know it, we're going to be into the spring. Although first, I teach virology starting in January. I'm really looking forward to that again. Questella's in Indiana. Nice to see you. Peter S. is in Boulder, Colorado. Leo, starting to get cold nights and mornings here in Mendocino. 59F, 14C, starting to burn the fruits of my chainsawing over the summer. Sandra, good evening. Nice to see you again. Visto from Sydney, Australia. Good day, Professor. And viral particulates, drizzly but warm, 21C in Sydney. Pete is in London, down to 10C, just got back from the shop just before the rain arrived. What is the shop? Is that a place where you build things, or it's just what you work, as you call it? I mean, the incubator could be a shop, right? Carol, our favorite nephrologist from Bakersfield, California, where it is, a bit mild, 22C. Logotic is in Chicago, 9C. Peter S. is in Boulder, Colorado, 18C. Miss Evelyn is here from who knows where. Privyet from Moscow. Veronika, 4C. Oh my gosh, cold and cloud. John is in Minneapolis, 6C. A little chilly there. Eileen is in Portland. Costella says we went from 85 to a freeze warning in 2 days. Maureen Mitchell is in Northeast Ohio. Miss Evelyn is in Hungary. Whoa, that's cool. Nice to see you. 8 degrees C. Joseph is in Southern Ontario, Canada. Pete says, I know how long I've been following MicrobeTV. Vincent is on another edition of Principles. So this is going to be the 6th edition. It won't come out till 2027. We've almost finished rewriting the chapters. We're working on redoing some of the chapter figures, and then it has to be, the proofs have to be typed out, set and type. Then we have to check them, and then it will get published. So most of next year will be proofing. But this will be the 6th edition, probably the last one we're going to do for a variety of reasons. But it's been a lot of fun, and I've learned a lot. No kings. Nope. We don't want any kings. Thank you, Kang. 50 years of communicating scientific truth. So I say 1975. That's the year I went into the graduate program at Mount Sinai. And then this is 2025. That's 50 years, right? when you can't read in real life, you make AI videos. So I have, full disclosure, I've been using AI to make some of the thumbnails for MicrobeTV programs. So for example, if we go to, let's go to the MicrobeTV website. We'll share this up. And let's get Visto off here for now. So let's go to, so this dementia vaccine, I made this image in one of the Google AI programs. I said, draw an image, or no, not draw. I said, create a photo of an elderly woman getting a vaccine. That's all I said. And there were originally two people in the background sitting there waiting. And I said, get rid of the two people in the background. And that's it. I mean, you can tell it's AI generated, but I could find something similar in a photograph, right? But this just works, so why not? And let's see, what else have I done with AI just to show you? Let's go to our podcast. Let's go to TWIV. And so there's that one. Daniel is not AI, of course. This one was done with AI, and this one was cool because these two phage pictures were all labeled in the original paper with, lines and letters and names, and I put them both into aistudio.google.com. Nano Banana is the model that I use, and I say remove all the letters and labels, and in 3 seconds, look, they're all gone perfectly. And when the lines go over the particle, they take away the color and make a white background so the black line sticks out. They fixed it perfectly. And then I said, put the two images on a television screen. And it did that. And you can see it's on the wall. And there's a little plant here. And here there's a little cabinet, which I sort of cut out. So it can be used creatively. And here's Daniel again. And I think that's the last two that I've done. It's Elka. She's real. This is an actual photograph of two chickens. So those last two, I have been playing with making pictures, right? So, they have a good use in some cases. But the presidential videos are disgusting. They're just amateur, juvenile, no taste whatsoever. But this administration. It has zero taste. Knocking down the wing of the White House to build a ballroom? My God. Hello, Abdurrahman from Brooklyn. Good to see you again. Jan is in Tucson, 87F31C. Patricia, Tropical Storm Melissa is active in the Atlantic. Be safe. Airy Ozone is in Asheville, North Carolina. Oh, Governor Newsom put up a video of himself crapping on the January 6th rioters, and yeah, he likes to imitate the administration, right? Jeff is in Elgin, Illinois, 8C and overcast. Dr. Leem is in Perth, Western Australia. Nice to have you again. Elizabeth is in West Virginia. Good turnout for No Kings. Mark Lamont is in the east end of Long Island, 14C. Beautiful sunny day after more than an inch of much-needed rain last night. Lise is in Chile, Columbus, 46. Good to see you, Lise. Christian is in Hershey, Pennsylvania. Mike is in Seattle, 14 and sunny. Jonathan is at Encinitas, 19C. Bowden is in Richmond, Virginia. James is in Longmont, Colorado, 19C. Barbtree is Durham, North Carolina, 17C. Aubrey, Richmond, Virginia, 12C. Has RFK Jr. allowed any new outbreaks, or are we in the midst of an epidemic that's being hidden? Well, that's a good question. You would hope that the press would pick up some epidemics. But right now, the measles is the big one. There's a pertussis outbreak. Influenza is ramping up. COVID will ramp up soon. And we'll look at the numbers because, he's discouraging vaccination. And so a lot of people are not going to get immunized this year. We'll see what it does. MN is outside D.C. EHS Talent writes that Dr. Fauci appeared in an interview a few days ago. Great to hear his perspective. You should invite him to a podcast. It's a good idea. And ask him what he thinks about what's going on and what's going to happen in the future. I don't know how to. I have an old email. I'll try and reach him there. Jefferson Spaces in Miami, 29C. Thank you, Barb Trees, for doing the questionnaire. So if you go to the latest TWIP episode, there's a link to a questionnaire about how you use TWIP, and Christina's doing this as an academic exercise. We'd love if you took it. Dagfin was born in 1975. Oh my gosh. When I started out, getting my PhD, you're only 50, Dagfin. Oh my gosh. Yes, Carol is going to be getting shingles vaccine because it clearly prevents dementia. Because in the paper we did last week, shingles vaccine clearly impacts the development of dementia in an observational study. Remember, you can't prove anything, but you have a great correlation there on many levels. Hit the like button, folks. 98 people here, 57 likes. Just hit it. Just hit it. John writes, donated by the link on screen. MicrobeTV made me look, and the link is up here, microbe.tv/contribute above my left hand. Right here. Why do I have it twice? I don't need twice, right? Yeah, let's get the other one out. MicrobeTV. We don't need that one. MicrobeTV made me look good at my 30th high school reunion when speaking with my friends that became an oncologist and another attorney for IP and cancer research. Cool. Yes, you can tell that, Will says, you can tell the ballroom never got any planning permission. Dwarfs the White House. It's ridiculous. Yeah, I saw a great cartoon on Instagram. There's a lady sitting in the doctor's office on the table, and the doctor is putting an x-ray into the machine, and he says to her, since your insurance won't pay for an x-ray, I'll show you an x-ray of the new ballroom. Right. Gallbladder is in Chatham, Massachusetts, where it's 58. Barbara's in Ithaca, 45, and windy. You know, Barbara, that's my old stomping grounds. Went to college in Ithaca at Cornell. Pete says, well, I'm still getting used to having a king rather than a queen. We could probably do with fewer princes. Eric says, Fauci with Offit. That would be interesting, wouldn't it? Privyet Elena from Reno, Nevada, 17C and cloudy. So happy to be here. Nice to have you. Tractor Poodle, Bangkok. Wow. 26C and sunny. Bangkok, where I understand I need to visit. Jason, greetings to you all from the country formerly known as France. Where'd France go? Says Christella. Well, maybe Jason is not happy with what's going on in France. I don't know. All right, so how about a book? I take this from Les. So Les said I'm reading this. So let me tell you what I'm reading. So I read on my phone, right? I have the Kindle app on my phone, and I read on the train. I don't always read. Sometimes I think. Sometimes I sit quietly and look out the window and think. But sometimes I want to read, and so here is what I have been reading. It's a science fiction book by William Gibson. I don't know if any of you have read William Gibson, but it's called The Peripheral. Now, I've read many of William Gibson's book. This one was published in 2014. It's a very cool story, which is summarized here in this in this Wikipedia page. It's actually a very detailed summary. I don't want to read the whole thing, but it says it's called a science fiction mystery thriller set in near and post-apocalyptic versions of the future. The story focuses on a young rural town American woman, her name is Flynn, who lives in the near future, and on a London publicist who lives 70 years thereafter. And they can communicate because, well, you'll have to read the book. But it's a very interesting idea. So the Flynn, she exists in what is more or less the present, and the world is in trouble because humans have trashed it, and they're about to undergo an apocalyptic event, which will lead to the future, 70 years in the future, where people exist. And it says here, a series of not quite cataclysmic events that have killed most of the world's population, leaving behind a monarchic class of gangsters, performance artists, and publicists in an otherwise deserted London. And they can communicate with Flynn back 70 years previously. Kind of interesting. But I have to say, the first third of the book, I had no idea what was going on. I don't understand the language. I don't understand the language. The words he uses, the phrasing and stuff. I'd be interested to know if anyone has ever read this, what people think about it, because it's weird. But now, so what happens is Flynn, she plays a video game. So supposedly her brother was contracted to play this video game. Everybody's into video games there. And he can't do it, so he lets her do it. And she apparently witnesses a real murder. And so the people in the future find out about this, and they reach out to her. And they want to find out who the murderer was, because she's the only person that saw it. And so she can't actually go to the future, but they have a technology where you put a cage on your head and your persona is projected to a peripheral, basically a robot that has your personality in it. And you can walk around, you can see the future in there, but your body is back in the past. A very interesting idea, right? But God, did I have trouble understanding this, because it's just written in a way that I don't get, with a lot of jargon and so forth. Anyway, thank you, SRR, for your contribution to science communication. I love science fiction, by the way. Especially, I think William Gibson is very creative and very unusual. And I've read most of his stuff. And there's another book after this called Agency, set in the same world. I'm looking forward to reading that. Patricia, thanks. Welcome. Good to have you again. Legotic thinks William Gibson may be our greatest living writer. Maybe. Kang, I got my Shingrix vaccine because of Amy's story of her aunt getting shingles infection in her eye, and nice to know it reduces the risk of dementia. I've also had both of my shots. Remember, you need two. Two gives you better protection against dementia as also. Tom writes, The Peripheral, a very good book, one of Gibson's trilogies. Don't give too much away. No, that's all I'm going to say. So what is, you got Peripheral, Agency, and what's the third? I don't know what the third is. I want to read them all. Will says, here in China, books in English are hard to get. I hate Kindle versions. I use Audible and audiobooks, and I'm looking forward to listening to Peter Hotez's latest book. I like the Kindle because I can travel and I don't have to carry a lot of books. You know, in the old days, I would carry heavy books with me, and now I don't have to worry about that. Razor, Jin is reading Zen and the Art of Motorcycle Maintenance based on some recommendations from office hours. Yeah, I recommended it a while ago because I just finished it not too long ago. Jason, thanks for the Gibson link. Do you know Ubik by Philip Dick? I don't. No, I've read some of his other work, but not that. Let's see what else I have here on my shelf. You remember I did a video a long time ago of what's on my bookshelf? I don't know if you ever watched it, but it was funny. I stood in front of the shelf and pulled books out. Yeah, nothing jumps out. MicrobeTV, that's me. Any thoughts on how Asimov's Foundation trilogy was reworked? I don't. I have no idea. I read it not too long ago. I loved it. The first book I really loved. The others, less so, but still interesting. And I didn't see that they made some kind of version, video version, right? I didn't see that either. There's also a video version of Peripheral, right? And I didn't see that. I wonder if anyone has seen that. But not too many people seem to have done this. So, okay, let's do some news here. Not a lot, but a couple of things I want to tell you about. And let's see. Oh, yeah, this one is important. As you know, this is a morbidity and mortality weekly report. So as you know, the staff of this publication was all fired last Friday in another CDC massacre, and then they were rehired because it was a mistake. Because the administration is so dumb, they don't even know who to fire or not. They make mistakes left and right. I have zero confidence that they can do anything proper except in their own interests. It's just, you want people who know what they're doing, and they know the opposite. They know nothing. Anyway, this is an interesting article in Morbidity and Mortality Weekly Report. Interim effectiveness estimates of 2025 Southern Hemisphere influenza vaccines in preventing influenza-associated outpatient and hospitalized illness. So this is from March to September 2025. So this is basically the Southern Hemisphere and their winter, which precedes our winter, right? So we can get an idea of how well the influenza vaccine is working, because it's most likely that those strains are going to come here for our winter. So just to briefly summarize, they're looking at vaccine effectiveness against influenza-associated outpatient visits and hospitalization. And they find that in the Southern Hemisphere, seasonal flu vaccination reduced influenza-associated outpatient visits by 50% and hospitalization by 49%. All right. And so the countries they looked at: Australia, South Africa, Argentina, Australia, Brazil, Chile, New Zealand, Paraguay, Uruguay. Those are the countries that reported patients, 42,000 patients. And this report is a deep analysis, but I've already told you the results, and it's about 50% effective at preventing hospitalization and severe flu. Is that good? Sure, it's good. 50 is better than 0. Could it be better? Sure. And hopefully we will get better at some point in the future. Is there a problem with flu vaccine? No. There are no serious adverse events, no major serious adverse events. You're going to get pain at the injection site. You may get fever, malaise, lethargy, but it's not as bad as getting influenza, which could kill you. So maybe there's a good match between those strains and our vaccine strains, and hopefully that will translate into less flu. But because we have RFK Jr. actively discouraging vaccination in this country, there are going to be many people who say, I was never so keen on flu vaccine anyway, so I'm not going to get it. And I would guess, and I'm going to predict, I'm not a good predictor, especially of the future, but I would guess we're going to have more influenza this year. Now, remember, we won't know why, because it's an observational thing, right? We're going to look and see how many cases there are. It could be there's less vaccination. It could be that the strain match with the vaccine is not very good. It could be other things as well, but maybe less vaccination. It could be studied. There could be a correlation with that. Okay, so that's a cool article. It's Morbidity and Mortality Weekly Report, September 25th, which you can get online. It's open access. You can see how these studies are done. Okay, That's one. And the second is a nice commentary in CIDRAP, why the CDC director is wrong about the MMR vaccine. So this is by Jake Scott, who is an infectious disease doc at Stanford, who's been on TWIV before. And, he reminds us that Jim O'Neill, the acting CDC director, posted on X calling for vaccine manufacturers to develop separate measles, mumps, and rubella vaccines to replace the combined MMR shot. So why did he suggest this? Well, because Trump asked for it. He said, I think we should give the shots separately. There's no scientific rationale for that whatsoever. And no monovalent vaccine, no monovalent mumps, measles, or rubella is licensed. Nobody is making them in the U.S. And so Scott says a very interesting, I'm sorry, I'm not screen sharing it. Scott says a very interesting thing here. This presents an unusual situation. The head of the federal agency responsible for disease control is advocating dismantling a vaccination program his own agency credits with eliminating rubella and reducing mumps incidence by 99%. Just this January, the CDC's website celebrated the 20th anniversary of rubella elimination as one of our greatest public health success stories. And so, first of all, this makes people uncertain about getting MMR, having their kids get it, right? So there's going to be less immunization. And the second is, if you want separate vaccines, for which there's no reason why we should do this, and we shouldn't do it. MMR is great. You would have to have a company conduct new clinical trials and get FDA approval for 3 products. 3 products. And that would take many years to do. But, combination vaccines are better. And he goes through the data here. A 2017 study found that 69% of children who received combination vaccines in general completed their full vaccination series, where 50% of those parents whose parents chose single vaccines, right? So you get a 19% improvement in vaccine compliance when you have combination vaccines. And, you have to keep vaccinating against these viruses. And he gives lots of examples of studies for that. For example, in the U.S., we had a 1964-65 rubella outbreak because it was before the vaccine. 20,000 babies were born with congenital rubella syndrome. When a pregnant woman gets rubella in the first trimester, there's a 65 to 85% chance of fetal death or multiple congenital defects. These kids can have miserable lives and very costly in terms of health care. And this includes deafness, blindness, heart defects, and intellectual disability. And he goes on to some of the other problems. So this is a really dumb idea from a guy with orange hair who doesn't know anything about vaccination and just says, I have a feeling we should do this, and being supported, as is the case with this administration, by a lackey who also doesn't know anything, the acting director of the CDC. You should all be appalled. You should all write your Congress people and complain that, and you could use this article as background, complain that we don't need the vaccine to be separated into its components. Oh my gosh. What a crazy world we're living in, right? All right, let's go back to where we were on the comments. So Eric says, folks might like the Ministry of Time by Kellyanne Bradley. Thank you. That's cool. That's cool. Firings are all a Drumpf intimidation ploy. Orobotodogo. Is it still worthwhile getting the shingles vaccine for someone who's had shingles twice? Daniel always says, this is Daniel speaking, not me. He always says, even if you've had shingles, you should get the vaccine twice. And the dementia studies are done, are looking at the impact of vaccination on dementia. So we don't know anything about infection. Well, infection is not alone, it doesn't give you immunity to reactivation, right? So you get infected as a kid, you get chickenpox, and the virus becomes latent in some of your peripheral nerves. Peripheral, William Gibson, interesting. And then after, generally after 50 years of age, you get reactivation and you get shingles, the rash. And it can also happen in your brain, and that's what leads to dementia. So you need a vaccine to prevent that, because when the virus is reactivated, the vaccine will neutralize it. So I would say in terms of dementia, based on that paper, you should get the shingles vaccine. It's a good question. Will writes, influenza is pretty low here in China, but we're surrounded by high surges in Japan, Thailand, Singapore, and now Hong Kong. He got jabbed a few days ago. Yeah, it's going to get there eventually. Marge is Amelia, Virginia. Starry skies and 13C. Markle, thank you so much for keeping us informed. I'm fading fast. We'll catch more of you tomorrow. Good night. Sleep tight. Don't let the bed bugs bite. If they do, take a shoe and hit them till they're black and blue. What a wonderful rhyme, especially as sung by Metallica, right? Lise writes, with the gap in info from the government, the New England Journal of Medicine and CIDRAP will be reporting an alternative to the MMWR. Yes, and Lise sent this to me today. I think this is a great idea because even though MMWR exists, you still have to be suspicious because many people have been fired, and who knows what can be done there at CDC. Carol writes, my hero, Stanley Plotkin, developed the rubella vaccine. Yes, which he talks about on Twiv. Elizabeth writes, this is Wakefield's fraud. He abused children, his patients, because he owned a patent for a separate vaccine. Want to bet one of Trump's buddies owns one of the patents? Indeed. Miss Evelyn asks, which vaccine or booster shot is recommended for people over 40? I don't know what disease are you interested in? Or are you interested in all of them? I don't know about 40 because the vaccines. So 40, the recommendations are flu vaccines, and in my opinion, you should get COVID vaccines as well. But the age is higher, and RSV vaccines is 75 and up, so you don't qualify for that. Shingrix is 50 and up. But you could write a question to Daniel at MicrobeTV and ask him that, and then he can answer it for you. Veronica got flu shot last week. Now I have to start looking for Sputnik. It's very difficult to find. Yeah, it's no longer rotating around the Earth. Just joking. I'm sorry it's hard to find. Shouldn't be. Tractor Poodle in Bangkok says that Shingrix is about $158 US. Yeah, it's expensive. I'm sorry about that. But it's good. It's good. Works really well against shingles and apparently reduces your chances of dementia. However, folks, let me tell you this. So Monday on Twin, this week in neuroscience, we did a paper showing that air pollution tracks with increased dementia. I'm not going to say it causes because it's an observational study. Well, part of it is an observational study where they looked at air pollution levels. And how do we know this? Because many countries sample air and they measure the pollution in terms of particulates, right? So much weight per cubic meter. I'm really scared that in the U.S. we're not going to be sampling anymore because that's done by the EPA. But communities east of the Mississippi have the highest air pollution, and west is lower, except for California, which is pretty high, certain parts of California. Anyway, they found a correlation between high air pollution and dementia, two kinds of dementia, Parkinson's and Lewy body dementia. But then they did experiments in mice showing you could feed. So these dementias are characterized by misfolded alpha-synuclein protein in the nucleus, forming these fibrils called Lewy bodies. And they feed this air pollution to mice. They don't feed it, sorry, they put it in their nose, so they inhale it. And the mice develop dementia. They develop these Lewy bodies in their nuclei. And they can take air pollution from 3 different parts of the world, I think China, Georgia in the U.S., and somewhere in Europe. And they can have mice inhale this, and they get dementia. And it's just so stunning and convincing. It's amazing. So you can get your Shingrix vaccine and, get a reduction in your chances of getting dementia. But if you're in an area with a lot of air pollution, you're screwed, because what can you do about it, right? You're not in control. What makes air pollution? Coal burning, fires, industrial activity, tires wearing out on pavement. There's so many things. Yes. Will says air pollution, it's high in China. Yes. And so it's very scary. It causes dementia most likely. I mean, I say most likely because, right, it's an observational study and it's a mouse study, but it looks really good and it can be confirmed by other people, right, which would be great. So we need to reduce air pollution, but in the U.S., EPA, the Environmental Protection Agency, is being gutted, so who knows what's going to happen. Vanity asks, I wonder if those toxic fume aviation events cause Lewy body dementia. I don't know. It's a good question. Lewy body dementia, by the way, is what Robin Williams had. He had Lewy body dementia because at autopsy, every neuron in his brain was full of these Lewy bodies in the nucleus. But of course, he killed himself because it was driving him crazy. He was depressed and he couldn't remember anything. And it's amazing he lived until his 60s. Let's see. Will an N95 mask everywhere against the particulate? I would guess a well-fitting mask should do that, right? Because this is particulate, which is much larger. Well, maybe it's about the size of a respiratory droplet, so it should be captured by the mask. I don't know the answer. It's a good question. And also, could an air purifier protect you in your house? It could, right? It could reduce the amount of particulates, but then you're going to go outside, and you're out a lot, right? And also, also dust particles, gas heating boilers can make air pollution. And as Veronica says, even without dementia, air pollution kills millions of people, right? Respiratory diseases and cancers, of course. So humans are screwed, folks. We made a world where we're screwed. That's The Peripheral by William Gibson. Writer Terry Pratchett had Lewy body dementia too. Lagotic writes, I've actually started carrying around an N95 in my pocket again for tear gas, though. I always have a mask as well for a KN95 for crowded places. Yes, John, Twin always has something interesting to cover. So that was Tim's pick. Very interesting. Elise writes, if you're looking for Novavax, our Costco and certain CVSs in Columbus have it. I tried Moderna and Pfizer's COVID jab, but personally found Novavax to have 0 side effects. Novavax jab this year. No, just aside here. I was doing the textbook today. We read the chapter on COVID, and the Novavax vaccine does not induce CD8 T cells. It induces CD4 T cells and antibodies, of course, and it's protective against severe disease, but it doesn't induce cytotoxic T lymphocytes, but the mRNA vaccines do, which is not a problem because clinically it's effective, but I just thought you might want to know. Lise writes, Nevitz. Ironically, reducing particulate air pollution leads to greater levels of global warming. The particulates in the higher atmosphere reflect sunlight back into space. You can't win, can you? I told you, humans have screwed the earth. Totally. Laura writes, coal burning releases mercury, lead, arsenic, PCBs, PCBOIs, dioxin, and radioactive biphenyls, according to the toxic release inventory data online from a coal-fired power plant. We should not be burning coal anymore. But I understand in China, they're still burning a lot of coal. And in many countries where did I read? In Mongolia, there's still some steam-driven trains in parts of the country, which are coal-fired, of course, because they don't have an alternative. They can't electrify. Oxygen therapy could help against what? The dementia caused by air pollution? I really don't know. It's a good question. Will writes, if the air pollution index is high, Chinese wear N95s outside. Our air purifier cleans it up indoors. Patricia writes, I think we need to work on better air purification and filtration in all of our buildings. I think that would be a great idea because building quality air is often not good. Yeah. Vanity writes, I love the curiosity you spark. Google says recent studies suggest potential link between exposures to air pollution from aviation fume events and an increased risk of Lewy body dementia. Interesting. Yeah. So you got to be careful when you go to those aviation events. Oh, my gosh. I've never been to one. Luis asks, would overprotection make it worse by not giving work to immune cells? Overprotection by vaccines? No. They give work to your immune cells. When you take a vaccine, your immune cells are working to recognize it and remember it. So that is not a problem. No. I wonder how much asbestos is in the East Wing of the White House. I'll bet they didn't do any mitigations on the asbestos if there were, right? They just knocked it down as fast as they could because that's what these people do who know nothing and who, if they happen to know something, they just cover it up. I would say that they probably just knocked it down and didn't even care. Pete Glass writes, a huge effect removing the sulfur from shipping bunker fuels. Meanwhile, the U.S. wrecked the IMO plan to decarbonize shipping last week. Not surprising. Aubrey wants to know, are there any quantifiers on the amount or nature of the pollution leading to dementia? So in different parts of the U.S., there are different levels of pollutants when measured in micrograms per cubic meter. And the risk of dementia increases with increased levels of pollutants. So you could look at the paper and get a number for a certain level of dementia, right? You can do that. But in the mice, they let them inhale 3 different amounts of pollution. They all led to dementia. Eric Jarvis, first time in L.A., 25 years ago. Smell. Thought bad thing in hotel room, cleaned up with a guess of chemicals. No, just what L.A. smog smells like. Smog from the Hollywood Hills. Yes, very Nika. We can win. We need to stop burning fossil fuels. But it's not just that causes the pollution, right? It's other things. But yes, that would be a big step forward. And really, China is pretty cool when it comes to lowering emissions. I don't know. I wouldn't know. I've not looked at it. But maybe someone who is there, Will is there in China, could tell us. would a super volcano eruption send enough particulates into the upper atmosphere to block sunlight, thereby circumventing global warming? I think that Mount St. Helens eruption did impact global temperatures for a while. And yes, I think that could happen. If you blocked enough sunlight, you could. Yeah, but then if you block sunlight, then plants can't grow, and this is a problem as well. Marg wants to know if ALS variants have an infectious trigger. A friend of mine recently diagnosed, but my friend is getting regular infusions and disease is not progressing. I don't know what the infusion is there. Fireworks make pollution too. That's right. You smoke all over the place. Coal burning in the Midwest spreads east into Pennsylvania, New York State, Massachusetts, and beyond, up into Canada. I thought Nova Scotia would be pristine. It's much worse than where I am in Connecticut. Julie says, is there any way to find out accurate flu and COVID data for October? Yes. So Daniel Griffin, in his weekly clinical report, has links to some accurate dashboards. Okay. I don't remember them offhand. One is a Johns Hopkins dashboard, and there are others as well. But if you go to the show notes, you can find that. Hello, Barb Mac, UK. It's awfully late for you, but welcome, another moderator. Clean energy does save lives. It saves grief, too, for sure. EHS Talent, what causes the arm pain post-jab for a few days? The needle injury or a reaction to the vaccine liquid? Well, of course, the needle can cause some hurt depending on how it's done. But it's really the inflammation, which is the immune response to the protein components of the vaccine that's causing the pain, right? So immune cells are coming in, cytokines are being produced, and increased volume, pain, heat, and redness, all signs of inflammation. Dagfin writes, we did see the climate effects for decades after the Krakatoa eruption. In the 1880s, temperature dropped globally, and sunsets were very colorful due to particles in the atmosphere. Oh, the 21 Balloons by William Pen Dubois. Did you ever read it? Oh my gosh, I got it here on my shelf. It's here. It's a wonderful account of the eruption of Krakatoa, which I guess properly now said Krakatau, but it was in the book, it was Krakatoa. Oh, yeah, because I was so fascinated by that eruption as a kid. Are there any studies on air pollution and any effect it may have on viruses, positive or negative to spread? Well, of course, air pollution makes people more susceptible to respiratory virus infections. Okay, so that is what I know. I don't know about any other any effects on the virus particles themselves, but the viruses could attach to particulate air pollution and can be carried along in air currents. I mean, we already know viruses are way up in the atmosphere and being carried along, so that could be an effect as well. But I'm not sure. Melba Moore writes, China has more population, therefore more constructions, therefore more energy to be utilized. So, no, therefore they pollute more than any other country. Laura writes, Hunga Tonga in 2022 was underwater, so sent up massive moisture into the atmosphere, took a bite out of the upper atmosphere. Okay, can tuberculosis exist in an animal? Is it related to the animal HPV? No. So tuberculosis, of course, is called by a bacterium called Mycobacterium tuberculosis, whereas human papillomavirus causes warts in humans, and then there are animal versions like Shope papillomaviruses in rabbits. So the question is whether the M. tuberculosis will grow in rabbits. Let's find out in animals. It does grow in a variety of animals, both domestic and wild. You know, you have bovine tuberculosis, cats, goats, horses, dogs, cattle, bovine. Can spread among them, but you know, humans can in some cases get infected from animals, but most of the human TB is human to human spread. And Barb Mack writes, our neighbor's cat had TB. It's a notifiable disease in the UK. There you go. Aubrey writes, during the COVID pandemic, I recall a dramatic change in pollution leading to restoring the view of the Himalayas. How interesting, right? Less anthropogenic activity. Mark says, thanks. Sorry I'm late. It's all Tylenol's fault. Eric writes, much value of MicrobeTV podcast is relaxed conversation between multiple experts, much better than one-on-one interviews, explores more stuff. I think that's one way to look at it, yeah. You get a variety of different viewpoints, right? And I like that, but I also like doing one-on-one. I think that's fun also. Yeah, pollution in India is a major problem. What country has the most air pollution? Chad has the worst air quality, followed by Bangladesh and Pakistan. That's from 2024. So the pollution we're talking about is particulate matter 2.5, PM 2.5. And Chad had a concentration of 89 micrograms per cubic meter, which is 18 times higher than recommended by WHO. And, you know, I don't remember the numbers from that paper, but they were in the same ballpark. Can human infants catch parvovirus from dogs? Let's find out. No, species specific, that affects only dogs. And Mycobacterium, according to Mark, maritimus will grow in fish. Albert says, no question, but wanted to express my appreciation for your incredible podcast. I've learned so much and love the incredible breadth of topics covered. I'm glad you like it. Thank you very much. Any plans for the domestic feline ID office hours? Yeah, I'm still pondering over that. It'll happen. Don't worry. You know, infections of cats. Eileen writes, TB is thought to have been brought to America by infected seals prior to Europeans arriving. Jim Joe Dudes writes, the RSV vaccine was lowered to 50 or 55 now with a pre-existing. Just tell the pharmacy have everything, overweight, diabetes, immunocompromised, then it's covered. Here we go. Dorothy, on the West Coast, there was TB in seals that spread to some Western tribes before Europeans arrived. Invite Heather Cox Richardson to talk about the history of public health in the U.S. That would be interesting. I read her column every day. And John writes, I remember hearing China led the world in installing new solar in 2024. So maybe some hope via renewables. Marge writes, the pandemic lockdowns were hard for many, and folks fuss about them a lot, but they really showed what the world could be like if our species could behave differently, even temporarily. Yeah. Yep. Will writes, our air purifier brings N2.5 down to 1 microgram. That's so that particle that I was talking about. Yeah. Ari Ozone is taking a class at UNCA taught by emeritized scientists on role of science in Project 2025. So challenging to sit through. A recent class looked at 25's goal to cut EVs and kill charging stations. What a just so stupid. What does Elon Musk think about that, right? I mean, this is also screwed up, not well thought out. Aubrey writes, I recall hearing the eruption of Mount Pinatubo in the Philippines offset years of global heating by reflecting solar radiation, but it only took a few years to return to previous atmospheric conditions. Okay. E. humerus, I find genetic resistance to disease such as the HBS gene conferring immunity to malaria with one copy heterozygous fascinating. Yeah, totally, and there are many more examples, right? And Laura writes, mercury is an immune system toxin. PFAS is an extreme immune system toxin, immune hyperactivity, aka cytokine storms in response to getting diseases. We are now allergic to disease. Yeah, I don't know, Elizabeth, if Heather Cox Richardson has studied public health history, although, her columns do reflect familiarity with public health. But I mean, I could ask, right? There's also seal with TB in Peru, and West Coast was reported there are 3 seals with TB by Kirby. As an introvert, Sydney writes, the COVID lockdown didn't bother me at all. Some people I know who are extroverts, though, practically were climbing the walls. And Eileen asks if molnupiravir is still available, and yes, it is. Okay. I want to do a mini lecture now, and then we'll come back to your questions. Now, this is going to be a little challenging for you. It's the last of the mini lectures from my course. I will, of course, have other mini lectures that cover other topics, right? But it's the last one from the course. And this has to do with transcription and RNA processing. Let's make sure, Vinny, that you share the screen. Okay. All right. It's not Virology 431A. It's office hours. But anyway, what is transcription? So here's the Baltimore scheme. Do you remember this? It's a way of classifying viral genomes according to the path they take to make mRNA. Now, in case you don't remember, messenger RNA is an RNA molecule in our cells from which proteins are made. So ribosomes attach to mRNAs and they make proteins. So the mRNA specifies the protein. And here's mRNA in the middle of the Baltimore scheme here. And all of these are viruses with different genomes. We have double-stranded DNA, single-stranded, double-stranded DNA with gaps. And the copying of DNA to mRNA is called transcription. And it has to be double-stranded DNA. It can't be single-stranded. It can't be gapped. It has to be double-stranded to be able to be copied to make mRNA. And the RNA viruses, which are also shown here, they do not, the process by which they make mRNA is not called transcription. It's simply mRNA synthesis, because transcription was originally defined as the synthesis of mRNA from double-stranded DNA, right? So that's the process we're talking about today, how this works. And in cells that are infected with DNA viruses, which we're going to focus on today, you need at least one protein, a viral protein, sometimes more, to get DNA replication. And so that means you have to make at least one mRNA, sometimes many, before DNA replication can occur. So imagine a virus comes into a cell, and this is a DNA virus. It's SV40. The DNA goes in the nucleus, and before it can replicate, at least one viral protein has to be made. So the DNA has to be transcribed, right? But as I hinted already in the Baltimore slide, not all viral DNAs are ready for transcription. What do I mean? That has to be double-stranded DNA. So SV40 DNA in the virus particle is double-stranded. It is ready for transcription. As soon as it goes in the nucleus, it can be transcribed. That's where the transcription occurs for most DNA viruses. Not all, but for most. And then the mRNAs can be exported to the cytoplasm and then translated into protein. Okay, but what about the viruses with DNA that is not ready? All right, so we have the hepatitis B viruses where the DNA is gapped. It's only partially double-stranded. It's got a piece of RNA attached. It's got a protein attached. This DNA cannot be transcribed, so it has to be repaired. And the host cell does that. When this DNA gets in the nucleus, the host cell does not like broken DNA, which is what this is. So it fixes it. It takes the protein, it takes the RNA off, and it makes it double-stranded. Now it's ready to be transcribed. The single-stranded parvoviruses, they're not ready to be transcribed. So the host actually looks at it and goes, I don't particularly like single-stranded DNA in my nucleus, so it makes it double-stranded. It's not genome replication. It's actually a repair reaction. So now the viral genome is double-stranded, and it can be transcribed. So the host cell unwittingly allows transcription of parvoviruses. And then, of course, retroviruses or RNA viruses, but a DNA copy is made of them, which then integrates into the host DNA. And then it is a substrate for transcription. But we already talked about that in the retrovirus lecture. Okay, so any virus with a double-stranded genome doesn't need conversion before it can be transcribed and before viral proteins can be made. That would be the adenoviruses, herpes viruses, polyoma, papilloma viruses, pox viruses, etc. Okay. Now, in the cell, there are 3 different kinds of polymerases that make RNA from DNA. They're called Pol1, Pol2, and Pol3. And the one we're going to focus on today is Pol2. This makes messenger RNA from DNA. It makes it for the host, of course, and for some viruses that don't encode their own polymerase, that host RNA polymerase 2 makes mRNA from viral templates. Okay, there are other polymerases that make ribosomal RNA and transfer RNAs, but we're not going to talk about that today. So here's the process of transcription. You start with a double-stranded DNA in the nucleus. So here's the nuclear membrane. That's nucleus on top, cytoplasm on the bottom. Double-stranded DNA, the RNA polymerase binds to the double-stranded DNA, begins to transcribe. The RNA is capped. We'll look at capping in a bit. And then you have a mRNA made. And many mRNAs have extra sequences in them called introns, which are removed by splicing. And then finally, we have an mRNA with a cap and a poly A tail that's been spliced. It's exported into the cytoplasm where it can then be translated into proteins. And then eventually mRNAs are degraded. They don't last forever. They have a half-life, which is specified by sequences in the genome, particularly the 3' non-coding region. And yeah, the mRNA vaccines, the mRNAs were engineered to have a certain half-life. So some genomes are actually exported with introns in them. Many viral genomes are, and we're not going to talk about that today. Now, transcription is regulated. Here's a DNA template, and there are lots of signals that regulate transcription. So here, this red arrow is the transcription start site. That's where the polymerase starts to make mRNA. And there are sequences that are called initiator sequences that regulate that. There's a TATA sequence upstream. There are local regulatory sequences and also distant regulatory sequences, all of which can impact transcription at this initiation site. And the site where initiation begins is called the promoter. Right? The initiator sequence is right at the start site, but the surrounding area, about 20 to 35 base pairs, called the promoter. It comprises the initiator and a TATA region. And this whole region here is called the transcriptional control region. And these are specific DNA sequences that bind proteins that regulate transcription. That's why they're all in different colors, because different proteins bind to this. So transcription is always regulated because you don't need to be copying a DNA into mRNA all the time. You don't need every protein all the time. So here's an example of transcriptional regulatory sequences in viral genomes. Here's a promoter. Remember, a promoter is a sequence where transcription begins. So here's the promoter. This is actually all the promoter region of the adenovirus major late promoter. You can see the initiator sequence, the transcription start site, and binding sites for different proteins that regulate transcription. Here's SV40, early promoter, again, multiple initiation sites and upstream binding sequences for regulatory proteins and other adenovirus promoters. You can see a lot of differences in the number of initiation sites and also the kinds of upstream regulatory sequences, because every virus has a different requirement. So the way the transcription works is a whole bunch of proteins bind to the DNA at the promoter. So here we have RNA polymerase, which is the enzyme, and all of these accessory proteins, initiation proteins, they all bind at the promoter. It then requires energy to get going. The polymerase initially makes some short transcripts and then finally gets going, copying the DNA. And here you can see the mRNA is coming out of the complex. And on the right here is just a diagram of how this works. So here's the DNA, double-stranded DNA coming into the enzyme. It's denatured, so you have one strand in the active site, and that is now the template for synthesis of mRNA, which is shown here in red. And the mRNA comes out of the exit channel of the enzyme, and then the DNA is rehybridized and comes out a separate channel as well. Okay, so that's transcription, copying of DNA by this enzyme. And here's just a cool example of how distant sequences can regulate transcription. Here's a RNA polymerase II bound to a promoter. And 10,000 bases away, there's a sequence called an enhancer that can turn up transcription. And it works by recruiting enhancer binding proteins. The enhancer is this orange part. It recruits enhancer binding proteins that then bind to the transcription complex and enhance transcription, probably by stabilizing it. Now, other proteins regulate transcription, right? We have DNA binding proteins that we've mentioned that bind at specific sequences, but there are also what are called viral coactivating molecules. They don't bind DNA, but they do modulate transcription, and many of these modify the activity of nucleosomes. So DNA in your nucleus is wrapped around proteins called nucleosomes. And if the DNA is very tightly wound around the nucleosome, which is shown in brown here, looks like a cake, it will not be transcribed, or the transcription will be low. If the DNA is loosened up, imagine loosening a thread around a spool, then it can be more easily transcribed. So how do you loosen DNA around a nucleosome? Well, you can chemically modify it by adding a methyl group or an acetyl group to the histone. It's called histone methylation or acetylation, and that regulates the transcription activity of DNA. These DNA binding proteins are modular. They have this is the schematic of one of these proteins. They have a region that binds DNA. They have a region that forms dimers because these proteins act as dimers. They have a nuclear localization signal because they need to get it in the nucleus after they're made in the cytoplasm, because the nucleus is where transcription occurs. And then they have an activation domain that interacts with the polymerase and gets transcription going. Okay, we have a quiz. Let's see who's been listening. What is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? Is it membrane fusion? A biosynthetic event. Making something. Membrane fusion, transcription, DNA replication, protein synthesis, all of the above. Which is it? Let's see. Vanity says B. And Leo says B. That's right. Barb says C. No, you need to make at least one protein before you can do DNA replication. And so to make a protein, you need to make mRNA. So it's transcription that has to occur first. So the answer is B, which a bunch of people got. Visto and Mark and Pete Glass and Nevitz and Luis all got B. So good. I'm glad you're listening. I don't mind if you don't, but, why come? It's okay. You can talk among yourselves. All right. One more thing I want to cover before we go back to you guys. Transcription is regulated, as I've implied, by these DNA binding proteins, by histone modifiers, right? And so virus genomes use regulation to make sure everything is made at the right time. And there are two kinds of regulation that we recognize. One is called cascade regulation, and there's going to be another quiz. So ChatGPT said B. They actually knew? That's funny. That's okay. They probably parsed my lectures. Cascade regulation. So we have a DNA genome, right? And we have two promoters. So the DNA genome gets into the nucleus of a cell. And the first promoter for gene X is active. It makes mRNA and it makes a protein. And the protein is made from the mRNA in the cytosol. This protein then binds to the promoter of gene Y and turns it on. And then you get mRNA of gene Y. So gene Y is inactive until gene X is active. And we'll see some examples of how this works in virus infections and why you'd want to do that. So it's a way of delaying the production of gene Y product until gene X is made. So that's a cascade regulation. And B, we have positive autoregulatory loops. It can also be negative, but this is a positive one. So you have a viral genome with a promoter driving the synthesis of mRNA for gene X. So this genome gets into the nucleus, it's transcribed to viral RNA, and the protein product actually stimulates the activity of that promoter for gene X, so you make more mRNA. So it's a way of dialing up the synthesis of mRNA. So it could be positive, this protein, or it could be negative. It could shut itself down after it's run for a while. Okay, so let's look at a couple of examples of programs, transcriptional programs in 3 different viruses. First, SV40, a small DNA-containing virus. It's got a small genome of about 5,000 bases. It's double-stranded and it's circular. So the genome is at the bottom here. It has an origin of replication, right? That's where DNA replication begins. It's bidirectional. We talked about that before. And there are 2 promoters. There's an early promoter, which goes to the left, and there's a late promoter that goes to the right. All right, so you make mRNAs in both directions, but when the DNA first gets into cells, you make only early mRNAs. So this is an example of a cascade regulation. So here's the timeline at the top of SV40 transcription. So the virus gets into cells. The early promoter is active. So that's transcription to the left here. So early promoter is made. One of the proteins that's made is large T antigen. And large T antigen activates the late promoter. And the late promoter encodes the proteins needed for DNA synthesis and the structural proteins as well. Let me redo that. The large T antigen stimulates DNA synthesis. And then DNA synthesis alleviates repression of the late promoter so it can be activated. So when the DNA first gets into cells, only the early promoter is active until DNA synthesis occurs, and then the late promoter is activated. And then the late promoter produces mRNAs that encode the structural proteins, right? So the large T antigen is made, it stimulates DNA synthesis, that stimulates late mRNA production, which encode capsid proteins. Now, why would you want to have this cascade? Because the idea is that until you make capsid proteins, until you make DNA by replication, there's no point in making capsid proteins because there's nothing to encapsulate. So you let DNA synthesis occur first, and then when you have sufficient DNA, then you start making capsid proteins to encapsulate the DNA. So it's a very simple example of cascade regulation. Okay. The late promoter activity is very interesting because normally it's repressed when the DNA first gets into cells. And so if you look at time after infection on the left here with the relative concentration, the viral genomes, right, they start to replicate in blue and the concentration goes up. And then as viral DNA replication occurs, the concentration of late mRNA goes up as well. There's a protein in cells called initiator binding protein, IBP. It is present at a constant level during infection. And IBP binds the late promoter. So when the DNA first gets into cells, the early promoter is active. But the late promoters here are bound up by this initiator binding protein, and it represses the late promoter. But as DNA replicates, you get more and more copies of DNA, and you effectively bind up all the IBP protein because IBP protein concentration is fixed. And as you have increasing amounts of DNA made, you eventually have molecules that have no IBP bound, and therefore that turns on the late promoter. So it's a really clever way of regulating the late promoter, right? Initially, it's bound by this repressor, and then as DNA synthesis occurs, the repression is alleviated, and that's why DNA synthesis activates the late promoter. Okay, so that's SV40. Here's a summary. So basically, the virus is taken up by endocytosis. The DNA gets in the nucleus. It's ready to transcribe. You make early mRNAs that encode large T antigens. The T antigen goes in the nucleus and stimulates DNA synthesis. That DNA synthesis alleviates repression of the late promoter. You now have structural proteins, structural mRNAs, or I should say mRNAs encoding structural proteins, and the proteins then encapsulate the viral DNA. So the function of the early and late phases is to delay synthesis of the structural proteins until DNA has been replicated. That's the function. And it's done by this cascade regulation. So adenovirus is the same idea, but a little more complicated. We have an immediate early, and a late transcriptional phase, right? Same as SV40, but an extra one. And so the adenogenome gets into cells. The immediate early promoter is active. It encodes the E1A protein, among others. That activates the early promoter, which encodes proteins needed for DNA replication. And then DNA replication relieves repression of the late promoter. Right? So again, the idea is you make the late mRNAs later in infection when you have viral DNA replication occurring. Now, the stimulation of transcription is very interesting, and this is something related to cancer that we talked about before. So E1A activates the early promoter. How does it do that? So E1A protein, this purple guy here, binds a cell protein called RB. So normally our RB is bound to this transcription protein E2F. And E2F is needed for the transcription of the early genes of adenovirus. And so with RB bound, the RB actually recruits histone deacetylases, which remove acetyl groups from chromatin and compact it so it can't be transcribed. So remember, chromatin is DNA wrapped around nucleosomes. In a compact configuration, it can't be transcribed. When it's open, it can be transcribed open or relaxed, and the addition of acetyl groups opens it up, and a histone deacetylase tightens up the chromatin and shuts off transcription. So the RB protein recruits histone deacetylases, and they shut off the promoters, the early region promoters. But the adenovirus early gene promoter, the immediate early, encodes E1A, which binds RB and prevents this repression of transcription. So now the E2 promoter is active. And remember from our discussion of transformation, if cells mistakenly continue to produce E1A, the cells will keep dividing because these promoters also in the cell control cell division, and that leads to transformation and eventually tumors. And here's the adenovirus genome to show you how complicated the transcriptional program is. Here's the double-stranded genome. We have immediate early mRNAs made on the left here. We have early region mRNAs made on the other strand in the other direction, and they encode the DNA binding protein, the DNA polymerase, everything you need to replicate DNA. And then the late promoter is on the top again, and that encodes all of the structural proteins. And so a more complicated regulation of transcription than in SV40. So here's what happens in adenovirus-infected cells. The virus enters the cell. The DNA is deposited in the nucleus. The immediate early genes are transcribed, and they encode, among other things, E1A, which goes in the cell and activates the early region promoter. So now you can make the mRNAs encoding proteins involved in DNA replication. They go back in the nucleus. The genome is replicated. And then finally, after DNA replication, the late region promoter is activated. You make mRNAs encoding the structural proteins, which go back in the nucleus and encapsulate the viral DNA. Amazing. It's just a coordinated dance that works so perfectly. And the last one is herpes virus. Similar control as adenovirus, but a twist that I want to tell you about. So herpes virus DNA has an immediate early promoter, an early promoter, and a late promoter. The twist with adenovirus, with herpes virus, is that the immediate early promoter does not work in a cell unless a viral protein called VP16 is present. VP16 binds to this immediate early promoter, and it allows the cellular transcription machinery to recognize it. Now, where does that come from? Well, actually, the VP16 is in the virus particle. So when the virus infects a cell, it delivers not only the DNA, but VP16. That activates the immediate early promoter. It makes proteins that activate the early promoter, which encodes replication proteins. DNA replication then ensues, and that alleviates the repression of the late promoter, which encodes mRNAs, which transcribes mRNAs that encapsulate the virus particle. All right, so here's a summary of herpes viruses. The virus binds to the surface. It's uncoated at the surface. The capsid moves down microtubules to the nucleus. The DNA gets in the nucleus. And you can see VP16 is coming out of the particle, and it's imported into the nucleus so that immediate early promoter can be active, which encodes proteins needed for the early promoter activity, which encodes mRNAs for proteins involved in DNA replication. And then once DNA replication begins, the late promoter is activated, which makes mRNAs encoding structural proteins. So this is why transcription is regulated. 3 different examples. So here's a quiz for you. Adenovirus E1A protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenovirus 4A2 and L4, is an example of A, a negative autoregulatory loop, B, repression of gene expression, C, cascade regulation, or D, Dimerization. Leo says C, and that's the right answer. John C, Vanity C, Visto C, Dagfin C, Nevitt C, Pete C, Mark C, Makomali, and Thomas says K. There's no K, dude. Louise C, C, Lori C, Elizabeth C, Les C. Yes, so everybody got it. It's C. Okay. Now, next time is not viral DNA replication. Next time is transcription 2, part 2, which we didn't cover today. Okay. There you go. There's your mini lecture. And again, next week will be the last virology course-based mini lecture. So let's go back to your questions for our last half hour here. And now I was smart enough to mark the last one. You Humorous writes, I'm a traveling nurse by profession. Hospitals are much scarier places to work now, thanks to RFK. Are there places in the country I should avoid? Well, it's hard to say. I mean, the places where there's a lot of measles, I would certainly avoid unless you've been immunized. But otherwise, everything is, I think, pretty much the same. Now back to the emissions. Very Nika writes, we can't rely on volcanoes to solve our problems because the main problem is our CO2 emissions. Humans need to lower emissions drastically and do carbon sequestration. So in this Peripheral by William Gibson, they have these big carbon absorbing machines in the future that make everything work. It's incredible. Mike writes, Trump's brain must hold a record for misfolded proteins. So actually, it's a good point. So Alzheimer's, Parkinson's, Lewy, dementia, they're all probably diseases called by, and prion diseases, they're all caused by misfolding of proteins, tau proteins, amyloid, prion protein, and alpha-synuclein in the case of Lewy body dementia and Parkinson's dementia. So proteins misfolding. A really interesting commonality. Pawasan, I haven't forgot. I just wanted to make it right, and I didn't have a chance this week to do it. We'll do it next time, I promise, okay? Next week. Yeah, I'm here next week. Try the veal. Yeah. Eric writes, the measles vaccine, the attenuated vaccine, has caused 2 deaths and 6 serious illnesses. But remember, this is out of millions and hundreds of millions of doses that have been administered. So overall, it's very safe. And the lesson is that people with immune deficiencies should not receive live attenuated vaccines, right? So as Eric says, they had severely deficient immune systems. But, if we could detect that early enough, we don't give them vaccines. And these were examples where they unfortunately did because the immune deficiency wasn't detected early enough. Talking more about lockdown, Kirby writes, introvert, but I want to go outside bad during that time. COVID, it was test for rabbit heart model disease too. They had tested it. I don't know what you're talking about. I'm sorry. But yeah, I understand wanting to go out. Humorous, the hospitals provided me with far more socialization than I wanted during COVID lockdown. I was jealous of the people who didn't have to go to work, even though I'm an extrovert. Will is an introvert, so not troubled by lockdowns, though Changsha never had a lockdown, only in specific cities with big COVID surges. Yeah, I guess I'm an extrovert, and I didn't mind working right here during the lockdowns. Right here, I used to teach my course, and I did 3-4 podcasts a week from here in my basement. Why does herpes zoster cause dementia? Not sure. but also herpes simplex. So one of the ideas is that, the virus is in the brain, and an immune response is the production of amyloid protein, beta amyloid, which is supposed to be a defensive protein, but it misfolds and makes deposits or plaques, and that leads to the dementia. That's one of the working hypotheses. Visto Tutti asks, can us eukaryotes do without single-strand to double-strand repair? No, we can't, because that happens when you have, your DNA gets damaged from time to time. And it could be that parts of it are single-stranded, or during replication something goes wrong and there's single strands. So that has to be repaired. Otherwise, it's bad because the cell sees it and will kill itself. So there are sensing pathways that will sense single-stranded DNA and lead to cell death. So the cell repairs it, right? And if it can't repair it, the cell will die because it will lead to mutations. Right? So it's really toxic. Could a drug to block repair be antiviral? Yeah, it could be, but it would kill the cell too. Yes. Will says, I love your mini lectures, but I wish I understood them. I'm sorry. Just keep listening to office hours. That'll do. Oh, Luis. Love the Baltimore scheme. That's why I'm looking for a lab to do double-stranded RNA immunohistochemistry. I'm so sorry. I got your email. I didn't respond. I will get to it tomorrow. I promise. Goodbye, Leo. Have a good evening. Got my vaccine and I am smashed by inflammatory response. That says Viruses Luna. Which vaccine? Shingrix? Tell me. Okay. Mark says, I love these illustrations. Are there videos of any of these processes? I wonder if an EM videography could help make this better understood. I know people who say it's just a drawing. Yes, there are illustrations of DNA replication, of protein synthesis, of transcription, of many aspects of cell biology. You can find them on YouTube. Yeah, for sure. So Humorous has been talking about the hardship of working in healthcare during that time. And he says it's guys like Vincent who are the heroes, not me. No, I wasn't a hero. I just sat here doing podcasts. They stopped the thing that was killing everybody. Those are the people who developed the vaccines, right? And they were at NIH and various companies here and throughout the world. I just tried to get people to learn what was going on, me and my co-workers, my co-hosts of my podcast. But I wouldn't say we're heroes. Thank you very much. Nevitz, this is so cool. The virus forces the host to produce a transcription factor that targets gene Y. I wonder how many millions of years it took nature to figure this out. Yeah, so these evolution created all of this complexity, right? And you often wonder how long it took, how many tries did it take to get it right. Laurie says, for cascade regulation, does protein X have to go back in the nucleus? Yes. So the proteins are all made in the cytosol, right? And then they have to go back in the nucleus because that's where the DNA is. You can always think about it that way because mostly you'll be right. The proteins are made in the cytosol. The DNAs are made in the nucleus. So any protein that needs to get to the DNA has to get in the nucleus. Eric says, Twim suggestion, David Desmarais. NASA aims on the ecology and biochemistry of biofilm. Sounds good. Got it. I'm on it. John, how do the viral DNA and capsids find each other? They find each other delightful, John. That was a Dixon des Pommiers joke. They have there are sequences on the DNA, the viral DNA, that are recognized by the capsid proteins. So they're diffusing around the nucleus and they bump into each other and the signals make them attach. They're called packaging sequences. Are the promoter proteins specific to an exact DNA sequence? Pretty much, yeah. Those sequences that I showed in color on the DNA, they have specific sequences that bind specific proteins. And if you change the sequence, it doesn't work. So they're called sequence-specific DNA binding proteins. Yeah, it's cool. The virus dilutes out all of the repressor protein. It's very cool. How sure are we COVID was zoonotic and not lab? For me, 100%. There's no evidence for a lab origin. All the evidence is the market origin from nature. Yeah, they call it adenovirus because it was originally found in adenoids. Yes, it's not the only place it is, but that's a lymphoid organ that picks up viruses because it's a foreign particle. I think they find each other delightful. A humorist said, my sister is an electrician, so she named my niece Jules. Sometimes people just take their work home with them, I guess. Jules is a great name. I love it. Laura asks, is E1A viral tumors only? Yes. Not found in any non-viral tumors. Yep. Nevitz acetyl groups covalently attached to lysine on the amino acids on the histone, which removes the plus charge of the lysine, which makes the histone less attached to the negatively charged DNA. That's right. So that's how attaching a lysine loosens up the coiling of DNA around the histone so it can be easily transcribed. Does herpes cause dementia? Yes, there's good evidence that herpes viruses, herpes simplex virus cause dementia, as well as herpes zoster herpes zoster virus. So the chickenpox shingles virus. Varicella zoster. I was looking for the name. Varicella zoster. Okay, let's see what else we have here. TB asks, is fever impacting transcription? I don't believe fever impacts transcription particularly, no. I'm not sure what aspects of viral reproduction are affected by fever, right? We say that fever, high temperature is a defense against viruses, but I don't know what aspect of viral reproduction is impacted. There are lots of steps, right? I don't know which one is. Laura wants me to explain dimerization. Okay, so dimerization means that two proteins get together. So here's one protein, and here's a second. They attach to each other, and now you have a dimer, right? And they can be the same protein in two copies, or they can be two different proteins. That would be a thumb and a forefinger, an index finger, okay? And then we have trimers, where three proteins get together, and that's very common in spike glycoproteins as well. Yep, dimerization, two proteins come together. Barb trees is a CRA, but essentially unschooled in science. I appreciate how much I learned from MicrobeTV. I hope you learn as well. That's my goal, to give you a lot of information. I'm giving you my 50 years worth of virology experience. Would a mutated binding region silence the gene and be a recessive trait? Yeah, it would. So remember, the DNA sequence where the transcription regulatory proteins bind, that binding is dependent on the DNA sequence. If you change it, the protein won't bind, so transcription may not occur. It'll be low. But yes, the other gene, remember you have 2 copies of all genes. If the other gene is unaltered, then you're still going to still bind the regulatory protein to the promoter, to the DNA, and so it will still make mRNA. So it should be a recessive trait, unless, 2 DNA's worth of transcription is important for the phenotype. But mostly it's going to be recessive. You're absolutely right. Good job. Yep. Vanity writes, interesting bit to add about Lewy body dementia. Microglia, which clean up brain debris, help at first, but as the disease progresses, they become detrimental. Jonathan writes, in case anyone is in my area, Encinitas, California, and is interested, or our area CVS has Novavax. Get in your car and go get it. Elizabeth writes, have been really cool developments with carbon capture that look promising. The largest problem is scale, but there is hope. Cool. I mean, I don't follow this, but I'm sure it's amazing, carbon capture. Yep. Vista writes, maybe microparticle pollution encourages protein misfolding. It could. I don't know what the mechanism is. I'm wondering about that. Right? The particle has to get in the brain, so it's got to cross the blood-brain barrier, right? And I don't know how that would work because they're pretty big. I'm wondering if they get picked up in like macrophages and get brought into the brain and then have an effect. But we don't know. It's an interesting question. But yeah, so the outcome is protein misfolding. In fact, they made in this paper recombinant alpha-synuclein, right? They add the pollutant particles to it and it misfolds and makes fibrils, which then if you give to mice causes dementia. Oh my God, what a Koch's postulate this was, this paper. It's really beautiful. The twin will be out next week. Miss Evelyn asks, is there something new to cure herpes? No, not yet. There may be. So if you already have it, right now it's incurable. But people are working at trying to get the genome out of you. It's going to be a while. A vaccine is not going to help you, but so a shingles vaccine, you can have the shingles virus DNA in you. So normally it would reactivate and you'd get a rash, shingles. But if you have the vaccine, it will prevent the rash. So it's possible a herpes vaccine could prevent, say, a cold sore upon reactivation, just like the shingles vaccine does. But we don't have a herpes vaccine yet. People are certainly working on them, and eventually they'll come out. John writes, do all DNA viruses have to go to the nucleus? And Mark writes, I think smallpox stays in the cytoplasm. So yes, pox viruses and the giant viruses, Mimi and Mama and Mumu, Pandora viruses, and many others, they stay in the cytoplasm. So what does that mean? They have to encode their own DNA polymerase and their own RNA polymerases because they're away from the nucleus and they can't use the nuclear enzymes, so they have to encode them all. That is in part why they're giant and have big genomes. Visto tells us herpes is Greek for creeping vine, like the spots that follow nerves. Patricia writes, I have both texts, Principles of Virology, 5th edition. I go back and re-listen while reading the pages. That's cool. I like that. Luis writes, I've always wondered how mRNA translation is regulated since the UTRs are from non-human. Of viruses, you mean? There's not a lot of sequence specificity there. It's just a 5' end with a cap and then to certain lengths, absence of secondary structure. And and that will be translated, right? Now, if you have more or less secondary structure, that can regulate the amount of translation. But there's no virus versus human UTRs. You know, they both work. They use universal signals. Let's put it that way. Laura writes, any input on P53 action removing DNA damaged cells and how that factors into cancer? Okay, so here's how it factors in. It turns out that most cancers, human cancers, have P53 mutations. Why is that? P53's job is to monitor single-stranded DNA and unscheduled DNA synthesis in the cell, which means DNA synthesis happening when it's not supposed to. If it detects that, it will have the cell kill itself by apoptosis. Okay. A tumor is composed of cells that keep dividing without any control. That's why it gets bigger and bigger and bigger and just invades everything, disrupts everything, breaks off and metastasizes and gets bigger and bigger and kills you. So that's because P53 is mutated and it's not able to do its job. So it no longer can kill a cell that has unscheduled DNA synthesis caused by, well, so to get a cancer, you need about 12 mutations in different genes that make the cells divide forever. And so these are in genes that control mitosis, but also in P53 is one of them, because P53, if a cell started to divide continuously, P53 would kill it normally. But if you have a mutant P53, it doesn't, and it grows into a tumor. So what would cause a mutant P53? You inherit it from your parents: cigarette smoking, air pollution, chemical carcinogens. You know, all the cancer susceptibility factors can cause mutations in genes like P53 that are essential for preventing uncontrolled growth. It's really fascinating. If you go back to, so there's a mini lecture on transformation and pathogenesis in office hours, but also my lecture on that is really interesting because it talks about P53. Yeah, so HPV, herpes, adenovirus, even SV40 make proteins that antagonize P53 because when they replicate, they need the cell to be replicating. And so they knock out P53 to do that. The problem arises if those cells don't die and they keep making the proteins that antagonize P53, then they turn into a tumor. Like elephants in the tall grass, Dixon, I think, said that. Yeah, that's a Dixonism. And yes, life occurred through a process of bumping into each other over millions of years. That's absolutely right. Lori really enjoyed TWIP 262 reminiscing about Dixon. It was delightful. It was. Dixon was delightful. You're so much into virology. It's great. I would love to know how your non-virus activities and hobbies in life. At this point in my life, I have only related to MicrobeTV. I travel, I teach, and I do my podcasts. Yeah, I know I read, but that's not really anything unusual. I don't have any other hobby that I do. That's it. What you see is what you get. And I understand it's not ideal, but I really enjoy it. I really do. Thank you so much for your contribution to science communication. You humorous. You humorous. I really, really appreciate it. Questella, appreciate your more than 50 years of virology experience so much. Thank you. Thanks you so much. Dimerization is almost always non-covalent. This is an important feature because the proteins can unbind from each other. Yes. Sam Ahmed says, during my third year in uni, I had to retake my virology course. During my retake, I fell in love with virology and my course was supplemented by Vincent's podcast suggested by one of my profs. I hope, I'm glad you did, and I hope you're moving forward with your virology career. All right. Let's see. Type 1, cold sores. Type 2, below the belt. How does 1 and 2 know what part of the body it's in? It's all about delivery, right? I mean, type 2 is sexually transmitted. But can I just suggest how a type 2 might get to your lips without actually saying it? So sometimes it happens because it's fine. Type 2 can reproduce in the oral mucosa, but normally it's down there in the sexual area. Can you imagine how it gets up here? I'm not going to say it. You figure it out yourself. Got it. Vincent also likes funny cat videos on Instagram. If you're following MicrobeTV, yeah, that's social media manager, but I have my own Instagram account. I do like funny cat videos. I also like workout videos. I like wildlife videos. I like all kinds of videos on Instagram. But that's not a hobby, right? I read somewhere a few days ago that only about 16% of us habitually read books anymore. It's a shame. But you know what? Besides a shingles vaccine, there are 2 ways to minimize your risk of dementia. Use your brain and exercise. Okay? And I feel that I'm doing both. I exercise 6 days a week. And I don't do it as a hobby. I do it to stay healthy. And I read not just books, but papers, and I think about them, and I do podcasts with colleagues, and I think that keeps my brain active. So hopefully I can avoid it. I also, I tell jokes. I told virus jokes at the giant virus meeting 2 years ago, and I'm going again in December, and I'm going to do more jokes. Yes. Well, Patricia, the site of preference is the upper or lower nerves, but it depends where the virus gets in. Because if a type 2 virus goes here, it's gonna replicate. Yep. You can't do everything if you do what you enjoy for a living. So are happy every day. Yes, and I don't feel that I do it for a living. I do it as a passion. I know I have to earn money to survive, but I don't look at it as work. It's a passion for me. Lear said, I got it from Agnes, but he didn't say how. Tractor Poodle, working on your potential lectures in Bangkok next year. How to contact you. Vincent at microbe.tv. That would be lovely. I'd love to visit. Okay. I think that's it for tonight. I want to thank all of our moderators for being here. Barb Mack, Vanity Nutrition, Les, and Tom Steinberg. Thank you so much for moderating. Really appreciate it. Oh, you want to do your current favorite book? Sure, you can do that while I'm saying goodbye. We'll keep it open for a few minutes. You mean favorite of all time or just your current book? As you know, I'm reading Peripheral by Thomas Gibson. I want to read the rest of the trilogy. Thanks, everybody, for coming. I really appreciate that as well. We have 130 people and 134 likes. I cannot complain about that. That is just beautiful, folks. When are we going to be back? I try to do this every week. So next week is going to be October 29th, Wednesday, October 29th. 8 p.m. Eastern. We'll be back here for another round of office hours. We will do a mini lecture on Powassan. Actually, I was thinking of viruses you can get from ticks, which would include Powassan, right? That would be cool to do next week. All right, folks. That's another office hours in the can. I really enjoyed being with you tonight. Here's the heart. Look at that heart. It's right here. Right here. I enjoyed being with you tonight. A lot of fun. Be safe until next week when we return. Bye-bye, folks.